Hello, everyone. My colleague Dan and I are very pleased to be a part of this year's meeting. Today, we'll be presenting to you on our next liquid biopsy assay, an exome-wide liquid biopsy assay to study tumor dynamics. So first, a bit about our company at a high level, Personalis. Personalis was founded out of Stanford University back in 2011, and we're a leader in advanced genomics and analytical solutions for precision medicine and personalized therapies. We're located in Menlo Park, California, and we have a CLIA certified and CAP accredited laboratory. And our solutions span from research to clinical applications, and we built a very robust infrastructure to support the assays and the amount of data that we generate. We receive samples globally in support of major applications such as biomarker discovery for immuno-oncology and translational research, and also we support the neoantigen-based personalized cancer treatments as well. Now, as we move from these conventional treatment modalities to more and more personalized precision approaches, the information required to guide these treatments needs to be much more comprehensive in nature to capture the true biology of the tumor. So we begin to see this shift from these smaller scale gene panels that mainly focus on pathways in cancer and identifying mutations in the tumor that can be targeted to broader whole exome transcriptome approaches looking at all 20,000 genes to account for multiple biological systems. And this has been the focus at Personalis. Advances in our technology have enabled us to engineer very comprehensive assays, whether they're tissue or plasma based, to better understand the tumor and immune system interplay. So to further describe our approach and strategy, I'd like to first begin with describing our ImmunoID Next platforms for solid tumor profiling that works in combination with our Next liquid, our liquid biopsy solution. And our ImmunoID Next product is developed to enable comprehensive characterization of the tumor genomics, but also the immune-related components of the tumor microenvironment. And we do this from a single tumor sample and a paired normal germline specimen. And we maintain a very sample-sparing approach. We can work with just 4 FFPE slides to derive both the DNA and RNA, which we then use to interrogate multiple biomarkers. And the scalable approach enables the identification of biomarkers of today that you see listed here, like tumor mutational burden or looking at microsatellite instability status, but it also enables a path for novel IO markers as well. And by interrogating both the DNA and the RNA features from the sample, we can begin to combine multiple biomarkers and produce more meaningful composite outputs with increased predictive power. And this is a unique capability of the platform. It's allowed us to identify a composite neoantigen score. And this is beginning to be the trend in the translational space in an effort to better understand mechanisms of resistance and identifying patients who are more likely to respond to different types of therapies and drug combination. A more integrated, multidimensional approach is needed. And we also know that these samples are very critical, precious patient samples that have been acquired. And traditional processes have often required multiple samples to be sent to perform multiple assays to get the data needed in the study. So for example, perhaps having to send out a blood specimen to get the HLA testing of the tissue, a tissue section for expression analysis, then sending another tissue section for assessing mutational burden. And the result is that sometimes investigators have difficult decisions, maybe a trade-off about which particular assay they can run when there is very limited sample. Also, maybe dealing with complex data integration when you're working with several different platforms. So at Personalis, we've created a more efficient process from sample receipt to data delivery. We provide a dual extraction of the tumor specimen. We follow a unique library creation and then process the sample on our ImmunoID Next to produce very data-rich analytical metrics and multiple key biomarker outputs. So ImmunoID Next was really purpose-built for precision immuno-oncology and designed to be this singular solution, taking a single sample prep and working here to consolidate multiple biomarker assays into one. And ImmunoID Next, the platform, is powered by a very innovative assay design. First, by the depth of whole exome sequencing coverage, but also by our proprietary enhancement and augmentation. So we do a 300x mean whole exome sequencing of the tumor specimen But ImmunoID Next utilizes our proprietary ACE, or accuracy content enhanced technology, to provide augmented coverage of those difficult to sequence genomic regions like GC-rich regions and others. And we do this supplementation across all 20,000 genes to give you much more uniformity across the footprint. And I'll speak to this a bit more in the next slide. And this is then paired with our enhanced transcriptome sequencing of the tumor. We deliver 200 million total reads. This also utilizes our proprietary ACE approach, and we perform an exome capture enrichment protocol. But secondly, what's very innovative within the design is that we provide these boosts of ultra deep sequencing coverage in critical areas of importance in precision oncology, with this boosted clinical grade sequencing coverage across a set of 247 critical cancer related genes associated with both solid tumors and hematological malignancies. Now, this additional boost is what enables diagnostic capabilities from our platform. We also have boosted the TCR and BCR gene regions, and this allows us to investigate the TCR dynamics coming from the specimen. And additional areas that we have also supplemented in coverage and enhanced are the HLA loci. We provide a variety of HLA-related analytics, including typing, somatic variant calling, loss of heterozygosity reporting. And we also report out on the MSI status, as well as detect oncoviruses. So hopefully, as you can see, this really makes it a first of its kind assay. It enables characterization of precision predictive oncology biomarkers in both the tumor and the microenvironment, but has been very carefully designed with enhanced sensitivity and truly optimized for oncology purposes. And our ACE, our proprietary chemistry, forms the technological foundations for many of the assays and approaches we use at Personalis. And the issue or the problem with the standard exome and transcriptomes is that they often have gaps and inconsistencies in sequencing coverage. And this is something that we've recognized and addressed with our proprietary ACE chemistry. And so this shows you here a visual of a schematic of a representative gene. If you're looking at a standard off-the-shelf exome, you have what I like to refer to as peaks and valleys across the footprint. Maybe areas very sufficiently covered, maybe some others that are suboptimal, but you can also have completely missed exonic regions. And if a variant that resides there, maybe a potentially immunogenic variant, resides in those regions, they would be missed with the standard chemistry. And so this is what we've recognized and addressed with our ACE chemistry. So this is a visual of now what you receive with our data, the blue plus the green, where we really strive to give that more uniform coverage genome wide. So the gaps where you have no blue coverage with the standard approach are now filled in by ACE. And we do this supplementation for all 20,000 genes with ImmunoID Next to result in accurate assessment of biomarkers and the mutational landscape. So with that introduction of our solid tumor approach, I'd like to now introduce the next liquid biopsy exome scale assay. And as you'll see, our strategy goes beyond where targeted panels are today to provide a much more comprehensive view of the tumor heterogeneity using liquid biopsy. And so Next Liquid is a less invasive method to identify novel biomarkers, and we have taken a different approach than the traditional panel-based liquid biopsy assays and have developed an exome scale assay sequencing at high depth with an average coverage of 2000x across the entire footprint, but also including a significant sequencing boost of those 247 cancer-related genes where we sequence at 5000x. In keeping with the comprehensive theme of our assays, as I just described, we provide the full picture of solid tumor profiling with ImmunoID Next, but now in addition, we also have an exome-wide look of the plasma. And some of the key applications we're looking to enable from the plasma are understanding the biology at baseline, but also in the context of the evolving tumor dynamics in response to therapy, such as understanding how and why acquired mechanisms of resistance emerge, how biomarkers or mutational signatures can change over time, or neoantigens can change over time. And we believe the plasma assessment is complementary to the tissue biopsy in capturing that true biology and heterogeneity of a tumor. And when you're trying to build a systemic view of the disease, it's ideal to have both the liquid and the tissue biopsy because there are elements of the biology that you can only see with one or the other. So when looking for this full characterization of the tumor, the tissue remains important because you can assess important info about the tumor microenvironment, interrogate the RNA, look at TCR alpha and beta, other biomarkers. But in conjunction with the tumor, the liquid is also important because it's more representative of the inter and intratumor heterogeneity. And the assay that we've developed is configured to profile the tumor and the matched normal and the plasma as a set to identify somatic variants and de novo events from 50 nanograms of cell-free DNA. So we're using a tumor-informed approach with the current configuration. And because Next Liquid Biopsy is an exome scale approach, we cover many more genes from each of the key immuno-oncology pathways shown here compared to a panel-based solution. So we can explore a broader view of the tumor biology. We've demonstrated exceptional sensitivity of the assay, and we achieve over 95% sensitivity for SNVs and indels in our boosted region for a variance at a 0.5% minor allele frequency. We also have similar numbers exome-wide for variants at a 1% minor allele frequency and above. So with that, I'd like to now turn over the presentation to my colleague, Dan Norton. He'll be presenting more data for you from our Next Liquid Biopsy approach.
So thank you very much, Erin. And so to continue our discussion on performance, we now want to demonstrate how we establish that performance. So we performed a number of different studies, and the first one that I'm showing here is with the EACR reference standard. This reference standard is called the Sera-seq ctDNA mutation mix version 2. And within this reference standard, there are 25 known driver SNV mutations spread across 23 different genes. And those genes are found here on the right-hand side in this grid. In the experiment, we used 50 nanograms of cfDNA as inputs. And we created 3 different dilution groups, one at 2% allele frequency, another one at 1%, and the final one at 0.5%. And the reason we did this is we wanted to establish performance across different allele frequencies. For each dilution group, we created 3 technical replicates. And turning now to the results of this experiment, here at the top, we have our 3 different dilution groups at allele frequency of 0.5%, 1%, and 2%. And we color-coded them. So the 0.5% allele frequency dilution group is this bluish purplish color. And the 1% group is at this orange color, and the 2% group is this teal color. Down the left-hand side are the 23 different genes as included into the reference standard, and on the right-hand side are the 25 different SNV mutations spread across these 23 genes. Along the bottom is each dilution group's technical replicate, so allele frequency is 0.5%, replicate 1, 2, and 3. Dilution group of 1%, replicate 1, 2, and 3, and a dilution group of 2%, replicate 1, 2, and 3. As for our results, coverage across each different technical replicate and across each dilution group is relatively constant. So at this 4,000x coverage. In terms of the allele frequencies across each dilution group and each technical replicate, it is pretty much exactly where we expect it to be. So in the 0.5% dilution group, we observe a very tight band at 0.5%. And the 1%, we expect, again, 1%, and that's what we observe across all 3 replicates. And the same thing for the 2% allele frequency. We also observe very constant allele frequencies there. In each dilution group, we expect to see this purplish color, and that's exactly what we observe from one technical replicate to another. So generally, across each replicate, we see this purplish color, as you can see here on the allele frequency spectrum on the right-hand side. There were a couple of noted dropouts in the 0.5 dilution group. In the 1% dilution group, we expect to see this orange color, and again, that is what we see. And the same thing for the 2%. We expect to see the teal color, and that is generally what we see across all 3 replicates. So the takeaway from this experiment is that for the 1% and 2% dilution groups, we detected all 25 driver mutations for a sensitivity of 100%. In the 0.5% dilution group, we achieved an average of 94.6% sensitivity across the 3 replicates. And generally across all dilution groups, we achieved high and consistent coverage, as well as great correlation between expected and observed allele frequencies. Now, it's important to note that these genes in the reference standard are all found in the boosted genes as part of our DNA footprint. Those 247 genes as part of next liquid biopsy all have boosted coverage because we increase the probe density across those 247 genes. So this is a good representation of the performance across that boosted region of next liquid biopsy. But because this is an exome-wide product, we also wanted to characterize the performance exome-wide. So one experiment that we did to demonstrate that is using a cell media system. And we created the cell media system using a normal background of B lymphoblasts and tumor signal using the breast carcinoma cell lines 1143 and 1954. We collected the media and centrifuged to remove the cell debris. And we extracted the cfDNA from the media, quantified it and aliquoted it using 50 nanograms of cfDNA input across 3 technical replicates, as we did in the previous experiment. We did the library preparation and the exome enrichment and sequenced to product spec, which is at 250 gigabase per sample. So the results from this experiment are shown here on the right-hand side. So for cell line 1143, across the x-axis, we have different allele frequency bins from 0.5 to 1%, 0.5 to 5%, 1 to 2%, and so on. And as a function of those allele frequencies, we're also plotting here sensitivity. So what we see as we increase the allele frequency, our sensitivity also increases. And we see the same pattern or trend below in 1954, where as we increase our allele frequency across these bins, our sensitivity also increases. So we set our exome-wide SNV performance or sensitivity at 1% and above at 85%. So now turning to seeing how next liquid biopsy can be used in actual patient samples. We performed an experiment using 5 late-stage colorectal cancer patients, and we wanted to look at tumor heterogeneity. So across these 5 different patients, here in the gray circle are the SNVs from the tumor, and in the green circle are the SNVs detected in the plasma, and then the overlap are the SNVs that were found both in the plasma and in the tumor. So for example, in patient 1, we see 63 additional SNVs that we did not detect in the solid tissue. Same thing for patient 2, where we found 45 additional SNVs and so on. So this is a really nice demonstration of tumor heterogeneity and how applying next liquid biopsy can assess tumor heterogeneity. And panel B is just a breakdown of the percentage of SNVs found in the plasma and the percentage of SNVs found in the tumor. Next, we also wanted to look at how does next liquid biopsy compare when we're looking at targeted panels and what they're able to detect. So in a cohort of 9 head and neck cancer patients, next liquid biopsy was able to identify mutations in regions that would not have been detected using targeted panels. So in the left-hand panel, these are the plasma SNVs detected by next liquid biopsy at 1,334. And if we were to use all the targeted panels that exist in the marketplace today and concatenate them into one master targeted panel, in this experiment, we would only have detected 93 total SNVs. Turning our attention to a much more targeted region where we're looking at just now immuno-oncology related genes, next liquid biopsy was able to detect 31 of those SNVs across these immuno-oncology genes, whereas the targeted panels would have detected 16. So again, this is a good demonstration of the breadth of next liquid biopsy and the additional information that can be gathered versus looking or using a targeted panel. Another experiment, also in head and neck, looked at next liquid biopsy, or looked at SNVs using next liquid biopsy to detect key cancer-associated mutations. And in panel A, that's exactly what we see here, is just the somatic landscape across different patients and different genes, with a frequency of specific mutations found across each one of these genes. On the right-hand side is a demonstration of one patient in this 9-patient cohort that did not respond to checkpoint blockade therapy. And next liquid biopsy was able to detect a significant increase in the allele frequency on the HRAS gene post-therapy. So when we compare post-therapy to baseline, that's this T0, we see an increase of approximately 1% allele frequency of this mutation in HRAS to approximately 6% allele frequency in HRAS. This is a study that is ongoing, and more data will be disclosed shortly. There are many different types of study configurations that next liquid biopsy can be used in. In this example, at baseline, to get a full comprehensive view of the cancer in any given patient, we might sequence the tumor tissue using ImmunoID Next, both at the exome and transcriptome levels, sequence the matched normal using ImmunoID Next at the exome level. And then in addition to that, also sequence the plasma using next liquid biopsy at exome scale. Again, this gives us a really nice comprehensive view of the tumor mutational landscape at baseline. In subsequent time points from baseline, i.e. time point 1, 2, or time point n, we would just use next liquid biopsy to look at the tumor landscape and how that's changing dynamically in response to therapy. There are two important points here to make. The first one is that we can monitor SNVs or indels from previous time points and see how those SNVs or indels are changing in response to therapy. The other one is that we can also detect de novo variants in each individual time point. That is, variants that were not found in any previous time points, to see if there are any new mutations that may be emerging due to the therapy. If there is no liquid biopsy taken at baseline, this is okay. We can also detect or monitor variants from the tumor only, but then detect de novo variants as we go on. So we just wanted to give you an overview of next liquid biopsy. Thank you very much for joining our presentation. And for any more information, please contact Dan or Mark. You can see their contact information below. And please do not hesitate to reach out. Thank you.